coming from Spain, and I'm working in the Silicon Nanotechnologies company and university spin-off located in the Science Park of the University of Valencia. Our enterprise produces the mesoporous silicon, and silicon is an essential element. Its dietary intake can be seen on the slide. There are three main reasons about why porous silicon is very useful for pharmaceutical industry. The first one is the simplicity of its chemical composition. It's very easy to control its biocompatibility and biodegradability, and it can be adapted for the controlled release of many types of drugs. Mesoporous silicon micro and nanoparticles are the perfect candidates for the development of multitasking delivery systems. The great interest of that systems can be seen by the intensive research of that field. Multitasking delivery systems simply means that different parts of the systems are designed to develop different functions. The inner part of the porous silicon can be loaded with liposoluble compounds, peptides and protein, or cytotoxic drugs, and it also can be loaded or functionalized with magnetic or luminescence labels, and the external coverage of the surface can be used for beating biological barriers, for targeting specific cells or similar functions. Here we have an example of an advanced research developed by a prestigious Houston research group, and it is on that picture the studies of the kinetic uptakes of microparticles, mesoporous silicon, by endothelial cells. What does our company do is to produce mesoporous silicon in several formats. Some photos of our material can be seen on the slide. We guarantee several crucial nanostructuring parameters as particle size, porosity, pore size, and surface area. And we also guarantee the scalability of these properties. Here are some examples of what we do. We can prepare material with several hundreds of square meters per gram, and we can control the particle size distribution and the pore size distribution, and also the chemical surface composition can be varied from hydrophobic to hydrophilic. On the bottom of that graph, we can see some study of the protein absorption isotherm using our material. Typically, the drug loaded on the system can represent the 60 to 80%, and the loading efficiency of drug can be 10%. And here we saw also some protein release profile, and it can be varied from several hours to several months, easily adjusting the pore size and the surface composition. And the last graph is particularly interesting. Diabetic rats are intraperitoneally administered, and as it is shown by the progressive glucose lowering during the more than 5 hours, the glucose loaded in the porous silicon can reach the bloodstream and keeps its bioactivity. And that's all. Thank you.
Thank you very much.